Well, today I'm going to talk about the ancestral ghosts in your genome. Essentially, what your grandmother was exposed to when she was pregnant, in terms of environmental factors, may actually influence the disease you get later in life, and you're going to pass this on to your grandkids. Essentially, this is what you would call a non-genetic form of inheritance, directly impacted by the environment. And it's called epigenetic transgenerational inheritance. So I'll describe that. And this turns out to be a very significant role in where disease comes from, and in general, just regulating most of biology. So for the past 100 years, in science in general, particularly the biological sciences, there's been a concept, a theory, a paradigm, a dogma, that's pretty much predominant way we think about how things work. And this is called genetic determinism. The concept is that the DNA sequence, the genome, is the building block for biology. And it basically regulates directly what genes are on and off. And that then turns around and influences the functions of different organ systems. This then gives you the biology of the organism. When the DNA sequence has an abnormality in it, a mutation, then the genes are not turned on and off correctly. The physiology of the organ systems is abnormal, and you have a susceptibility to develop disease. But this affects all of biology. In evolutionary biology, the current concept is random DNA sequence mutations drive evolution. Pretty much, if you want to think about anything in science, we think that the DNA sequence is the driver. So what I'll talk about is, it turns out the environment has a major impact on biology. And for the past 50 or 60 years, a large number of observations have been made to suggest that genetics really can't explain many of the things we're seeing. The first is there's regional disease frequencies. Go anywhere in the world, the frequencies of different diseases is different. If you take someone early in life before the age of about 5, move them to a new region, allow them to grow up, they will develop the disease frequency where they grew up. So this can't really be a genetic phenomenon. It has to be more of an environmental phenomenon. We've looked at a large number of the major diseases we have, and we've looked for genetic mutations that correlate to those diseases. And in many cases, we have found genetic mutations, but pretty much across the board, it's in less than 1% of the population with the disease. Less than 1%. So it's good that we found some genetic mutations, but what about the other 99%? If you look at pretty much any of the major diseases, the increase over the past 20 or 30 years is so dramatic. In many of them, it's a 10-fold increase. For autism, it was 15 years, 10-fold increase. I'm sorry, there's no genetic mechanism known that could explain that phenomena. It has to be induced by the environment. If you have identical twins that have essentially the same genetics, allow them to grow up in different regions, guess what? They develop different diseases. If it was all genetics, they should have the same diseases. There's hundreds and hundreds of environmental factors that are known to promote or be associated with disease. The vast majority of environmental factors cannot change DNA sequence. They're not mutagens. So how is it an early life exposure promotes a later life event? In terms of evolution, there's these rapid evolutionary events that basically we can't really explain easily with classic genetics. So there appears to be an additional factor that regulates how the DNA functions. It's not the DNA sequence and the genetics isn't absolutely critical. It is. But it appears that there's something else that's regulating how it works. It basically goes forward. So what I'm going to talk about is epigenetics. This is what the phenomena is. I'll describe in a minute. Epigenetics provides solutions for a number of the failures for genetic determinism. Again, I'm not suggesting that genetics isn't absolutely critical. It's just a small piece of a much bigger story. Okay? So epigenetics are these small molecular factors around the DNA that regulates how the DNA functions, what genes are on and off. But it does this completely independent of DNA sequence. And so it's these molecular factors around DNA that regulate how it works, what genes are on and off, but it's independent of DNA sequence. And when a cell divides into two daughter cells, not only does it replicate its DNA, but it also replicates its epigenetics so that it keeps that, maintains that cell identity. So this is what epigenetics is. There's four main epigenetic factors that we know about. The first one identified was DNA methylation. A small chemical group called a methyl group gets chemically attached to the DNA. When that happens, it has the ability to turn genes on and off. The next thing identified was these groups of proteins called histones. Turns out DNA is wrapped around these histones like beads on a string. And when the histones are chemically modified, they can influence what genes are on and off, independent of DNA sequence. Another one is chromatin structure. Basically, whether there's a loop, a coil, or a twist in the DNA can also turn genes on and off, independent of DNA sequence. And the last one are these non-coding RNAs, these small RNAs that get expressed, and they come back and react on the DNA to regulate how it functions, independent of DNA sequence. So these are the four known epigenetic processes. I'm going to focus on DNA methylation because it probably has the most important role in programming events in terms of developmental biology. In terms of DNA methylation, there's two times during the lifespan of an organism where there's a major reprogramming of the epigenetics. The first one is during fetal development at the time of sex determination, when the fetus determines whether it's going to be a male or a female. The cell the precursor cell for your germline, whether you're going to get a sperm or an egg, that cell is demethylated. Methyl groups are removed. At sex determination, they're remethylated in a male or female specific manner, so you end up with a sperm lineage or an egg lineage. So this is the first time. The second time is at fertilization. So as you become an adult, reproduce, at fertilization, the sperm and the egg come together, and immediately within a cell division, the DNA starts to be demethylated. This is what creates, eventually, the embryonic stem cell that can turn into any cell in the body. Then as the embryo develops a little bit further, it will remethylate in a cell-specific manner. So this is a reprogramming of that epigenetics. There's a set of genes, though, that are protected from that demethylation. They don't get demethylated. They're called imprinted sites, and they basically maintain their DNA methylation, and they're protected from it. So thus, they can get passed from generation to generation. Thus, a non-genetic form of inheritance. So we did an experiment. We exposed a gestating female, what we call the F0 generation. So the gestating female rat, basically, that we expose is the first generation. We exposed it during that time of sex determination, just transiently, and removed the exposure then to a number of different environmental toxicants. We then bred the animals, those offspring that were born and grew up. When they grew up, it turns out they started developing large amounts of disease. Tumors, prostate disease, kidney disease, testis disease, and very high frequencies. So then we bred the animals again. So we had the grand offspring. We had equally high levels of disease in all the same diseases. Bred it again to the great grand offspring. This is the F3 generations, 3 generations removed from any exposure. The only exposure was in that F0 generation female. Also had high levels of disease. Took it even further to the 4th generation, same levels of high levels of disease. So we induced this, and it gets passed from generation to generation, even though the exposures are way back here. And the level of disease is extremely high. What we see in the males is predominantly testis disease, infertility. We have prostate disease. Those are specific to the male. In the female, we see ovarian disease, pubertal defects, and also pregnancy defects. In terms of both male and female, We see kidney disease, increased mammary tumors, behavioral effects in terms of anxiety and stress, and a large amount of obesity as well. 90% of the animals have multiple diseases. This is one of the highest frequency induced diseases ever found in any animal model. Okay. We've now repeated this with 8 different environmental factors: agricultural fungicides, agricultural pesticides, industrial contaminants like dioxin, plastics like bisphenol A, the insect repellent DEET that all of us use in terms of keeping insects away, the historic pesticide DDT, and also hydrocarbons like jet fuel or oil. Each one of them independently promoted these transgenerational inheritance of disease. Now, other laboratories have also shown that nutritional defects, both high-fat diets or caloric restriction, will do the same thing. In plants, temperature and drought did the same thing. Smoking alcohol is another factor that's been shown. And more recently, just in the past year, a number of different forms of stress, particularly maternal stress, will actually promote this as well. So this is a large number of different types of environmental sort of actions. This has now been shown in plants, flies, worms, fish, rodents, pigs, and humans. Therefore, it is very highly conserved. This is not a unique one-off just for studying rats. It really has been shown in most species investigated. So this is a complicated graph. I'll explain it here. So it turns out that the only cell that's going to pass any information to the next generation is a sperm or an egg. It has to be a germ cell. No other cells in your body passes anything to the next generation on a molecular level. Okay? So, to show that this phenomena was induced through epigenetics, which we talked about earlier, we took the F3 generation sperm, essentially, F3 generation sperm, and looked for epigenetic changes. So this is a complicated graph that basically shows all the chromosomes, 1 through 20, and the size of those chromosomes. So this is a snapshot of the whole genome, okay, all your DNA. And every single red arrow essentially is a differential methylation site. It's what we call an epimutation that's been induced. There's an abnormal differential methylation. So that's an epimutation. This this particular signature here was induced by the agricultural fungicide in terms of epimutations. There was over 200 of them. And this turned out to be an exposure-specific signature in terms of where all these things are. Or you can also consider it a fingerprint of your ancestors' exposure that's being passed through the germline for generations to come. All right? So, we did the same analysis on all those other factors we actually analyzed. Plastics, DDT, jet fuel, pesticides, vinclozolin. So you don't need to worry about this complicated graph. The only thing to pay attention to, zero overlap. What this means is each exposure had a specific signature or fingerprint that was not overlapping with all the other ones. So theoretically, we have a diagnostic now to say what your great-grandmother was exposed to during pregnancy, or what you're exposed to, and you're going to pass it on to your grandkids. This could be a major advance for us in the field of environmental sciences. We also know then which diseases these are directly associated with. So essentially, what I just described is an environmental factor affected the developing fetus during that time of development of the germline, the sperm or the egg, and the epigenetics got reprogrammed. As that individual became an adult, reproduced, in its germline, it's passing this altered epigenetics, such that the next generation then, the embryo now has an altered epigenome. Every cell that's generated from that embryonic stem cell now has an altered epigenetics and an altered set of genes going up and down. Those tissues sensitive to that will develop disease. Those that aren't won't. And so then basically as this one reproduces and goes to the next generation, it's the same phenomena, and this keeps going generationally. It's a non-genetic form of inheritance. This is just to show you one example. So what we did is we did the exposure with DDT. And when the exposure was just this transient period of pregnancy for the F0 generation mother, then we bred it for 3 generations, both male and female lineage. And what we found was there was a whole series of diseases in the F3 generation. But the most predominant disease turned out to be obesity. In the first generation, we did not see any obesity. But by the 3rd generation, half the population, both males and females, had a susceptibility to develop obesity. This is called a susceptibility. This phenomena doesn't promote the disease, it promotes your susceptibility to develop disease. So for example, if we had 2 individuals, one susceptible, one not, on exactly the same diet, exactly the same exercise, exactly the same living conditions, the one that's not susceptible wouldn't develop obesity, but the one that is will. So this is truly a disease that there's a susceptibility for. Diet and exercise is absolutely critical for the onset of the disease, but your susceptibility to develop potentially came from your ancestors. This is a completely different way to think about where a disease comes from. It also means what we're exposed to today is going to affect our progeny for generations to come. So this is a very different way to think about and a different mechanism for how disease develops. So, what I just told you is the environment has the ability to dramatically affect the epigenetics. This then influences how the DNA functions in terms of what genes are on and off. And these epimutations, these epigenetic epimutations that get put in place become a permanent constituent of the DNA. This then causes this change in what genes are on and off, and with organ systems, if it's a normal biology, then you're going to have normal biology going forward. If there's an abnormal expression and there's abnormal biology, you'll have a susceptibility to develop disease. So in terms of another consideration is, this doesn't only affect disease etiology. You also have an increase in phenotypic variation. The characteristics that the individuals have within a population are different. This has direct impacts on natural selection and evolution. Therefore, environmental epigenetics may play a significant role in evolution, which we hadn't considered before. So it doesn't just affect disease, it affects really all of biology. Okay? So essentially, as I started out, these are those ancestral ghosts in your genome, these epimutations that were put there through an environmental stressor. Now, what I just talked about is a pretty doom and gloom situation. What your ancestors were exposed to is going to affect your disease, and what we're exposed to, likely a different set of contaminants, we're going to pass on to our grandkids as well. Okay? However, so I'm going to give you the upside. So essentially, knowing this phenomena exists, knowing that there's epimutations in your genome that may be exposure specific and directly correlated with disease, we can now use these as diagnostics for those past exposures and potentially what disease you're going to get later in life. So early in life, in your 20s, we could potentially analyze your epigenome, determine what your ancestors or you were exposed to early in life. Because of that, what diseases you have a high susceptibility to get later in life. This allows us then to put into the whole thing called preventative medicine. We can do preventative therapeutics, preventative lifestyle changes, and so forth to actually prevent the disease from ever developing. The reason we haven't done that yet is we never had early stage diagnostics. Genetics has not provided that to us today. Epigenetics gives us that capability. So we may not be able to fix it, but we definitely can treat it, and this would have a significant impact on future medicine health of the population. That doesn't mean at all that we shouldn't clean up our environment, get rid of these toxicants, to get rid of these environmental factors. But during the interim, we have actually a way to maybe treat the condition and help the health industry and our healthcare. So with that, I'll finish, and thanks for your attention.